We're gonna record now. It did start recording, right?
Yep.
Okay. All right. So let me show you here the exercises tab. I split it into questions, assignments by date, and by the way, let's take a look at these assignments by date, because we're going to be following these lists here today. I think today it's going to be slightly different. We're going to rely a little bit more on the exercises, although we're going to cover the variant call notebook today. So as I was mentioning here, there's here the questions link. And if I click on that, you're going to see here that I have divided the exercises pretty much into the contents of the 4 weeks of the course. This is not entirely finished yet because I need to upload material here still. But towards the end of the course is where you're going to see that the exercises are hopefully more up to date. I still plan to do some work on this mini project, the impact of hypoxia on tumor microenvironment. Maybe I should include some figures here. And I think one of the figures, maybe one of the figures is this one, or one of the figures above, and then this figure here. And then I do not know if there's going to be any other figure here that is useful. I also have here some exercises about the genetic variants and haplogroups. And here this Wikipedia article about haplogroups, let me show you guys. It's pretty interesting. Here they discuss a little bit about what's a haplogroup. So it's a good idea, I would say, to read this article if you can, because I do not discuss so much about what a haplogroup is in here. But I asked some questions here, and these questions were thought according to the code notebooks. I hope it's going to make sense when we get started on these questions. What else? I have not worked a lot on the preliminary solutions, so I'm going to discuss as many of the questions with you on class, but I will not have the time, honestly, to update the solutions so much this week, which is our last week. What else? I want for the final presentation that you guys, again, you can use the commands. You can use commands either in Bash or in R, but please consider using the data about the mini project, and the mini project is the impact of hypoxia on the tumor microenvironment. I guess that's pretty much it, but there's going to be the survey on Friday. And I'm going to try to help you as much as I can for the presentations that we're going to have on Friday, okay? All right, let's move on here. So let's go to I'm gonna close this. Let's go to this notebook here, the variant call pipeline using sequenced RNA. And the reason why I called this variant call using sequenced RNA is because last week we did not have a session because it was a holiday, but there was a video. If you have not yet watched the video for this class here, you can. Nucleotide. So that video is about DNA sequencing. And that's the reason why I'm calling this variant call with sequenced DNA. All right, so let's get started on this notebook here. We are going to process mitochondrial DNA FASTA sequences from these self-declared white Brazilian individuals. These sequences were downloaded from the gene bank. We are going to process these FASTA sequences. And I'm saying here that we are going to align against the HG19 UCSC genome, reference genome. In fact, what we're going to do is we are going to align them against a one of the mitochondrial sequences, because unfortunately this reference genome is it's difficult to transfer it to the instance that we have right now because it's too heavy. So I gave up on doing that and using a mitochondrial reference instead. The end results of the variant call is a type of file called a VCF file. And here I'm mentioning machine learning algorithms, but we are not actually going to use the VCF files in a machine learning algorithm. So this figure here illustrates a schematic of a variant call pipeline. We begin with the sample sequences, and these sequences are the sequences that we downloaded from the gene bank, for example. Then we are going to do a round of alignment. We're going to do processing on the files. We will have the variants. Does anyone know another name for a variant? Does anyone know a name for a genetic variant?
Like any type of variant?
Yeah.
Like SNPs or?
Yeah, SNPs, SNPs or SNPs are one type of a variant. In general, you can also call a genetic variant a mutation. And the ones that we are going to mention today is the single nucleotide polymorphism variant, the SNP variants. Thank you, Atenesh. So after we identify these variants, and again, these variants are identified in these files called VCFs, which stand for variant call format. We can do some evaluation on these variants. And for us here, I do not think we're going to do this step. So we are going to end here with the identification of the variants. So the first step that we need to do is to process the FASTA sequence. We discussed briefly last week how the mitochondrial sequences, they're just this small. And that's wonderful because that allows us to work with them here, because if it was like an RNA-seq FASTQ sequence or a FASTQ file, we could still work with them here, but it would be much, much more difficult because those sequences are much bigger and heavier than these small FASTA sequences. And in this point here, if you read through this part, you're going to see that here I am discussing how the sequences here are lowercase. There are the spaces in between the sequences, and I did some processing with the sequence so that it became capital, all capitalized, and removed the spaces. And for whatever reason, I have not included an image to depict just that here yet. So this here, I think this is the exact code that I use it to make the letters go from lower to uppercase, and then I produced the FASTA file, right? This dot FASTA file, and I called it exercise because I prepared it for the exercise that we did last week. It contains multiple FASTA sequences and they are all capitalized. Now this part here, I have to deal with the name of the sequences. I'm not going to go into too much detail about this for loop here, but you can also see a for loop here. The way that this for loop works is it takes into account each one of the FASTA sequences that we have in a folder. And this folder here, in this case, it's called split_files. Maybe it doesn't make a lot of sense, but so here I am saying change directory to the folder containing all FASTA files. What this means is that I am going inside this folder in the terminal, and there I am running a for loop, which we are going to discuss in a little bit. And what that for loop is doing is it's oh, okay. What the first step here, for example, is doing, it is taking the header, which is the very first line, which means this line here, and it's using this information to rename the file. That's what I mean by rename here, okay? And then the reason why I put the part 2 here, the UCSC Hummingbird server, is because that's where the HG19 reference genome is located. So I have to do the alignment using the complete reference genome. And the reason for that is because the HG19 genome is so big that I only can use it in the server. And as I said, I didn't find it to be a good idea to run the alignment using the complete human reference, the complete HG19 reference genome. So what we are going to do for the variant call, we are going to copy the variant call folder that is sitting in the, or I already did that. I already did this. Sorry, I did it in this step. Here where I'm saying processed FASTA files are transferred to the Amazon image machine, which means I transferred it to the computer that we are going to use today. It's this computer here. Maybe it doesn't make a lot of sense now, but I hope it's going to make sense in a little bit. So together we are going to transfer this variant call folder to the Amazon image machine. You're not going to index. We are going to start there with aligning the FASTA sequences to the reference genome, which in that case is going to be one of the FASTA sequences. And so that alignment is very important. At least the idea of the alignment is very important because we are going to be able to identify mutations in the mitochondria sequences. This schematic here is more of a FASTQ file alignment because the FASTQ file is composed of several small reads, okay? Whereas the FASTA files that we are going to align today, they are a single sequence. And as far as I remember, the sequences that we are going to work with, they are 300 base pairs long, 300 nucleotides long. So they're not so big, and I'm very happy about that. So you can read this and you should really read all of this if you have a chance to revise this. And I hope it's going to make sense about what we are doing. Okay, this chunk of practice is going to be where the variant call pipeline is sitting. We are not going to do it right now. We're not going to do it right now because I want you guys to practice a little bit on the terminal. I want you to know that this is a variant call pipeline, and we are using a for loop strategy here. And I'm gonna say it, and I hope I have to repeat myself multiple times. We are using a for loop strategy right here because we are going to do something for all of the FASTA files that are going to be sitting in a folder in the server that we're going to use. This is one example of the results of the variant call pipeline once we run it in the computer. It's here just for illustration for you. And once we are done with running the variant call pipeline, we are going to have, as I said, the variant call format files, or the I guess it could also be variant call file, the VCF files. And what the VCF files are going to contain are the SNPs or the SNPs variants. These are genetic variants. These are very common genetic variants. And what they are is they are variation that we have in a certain that any organism that uses DNA as their code of life or their code for life is going to have some modification in a base pair when you compare individual one, or when you compare the purple individual to the blue individual to the orange individual. They have their DNA sequence exactly the same at this position, this position, this position, but then in this position, the nucleotide is different. And after this nucleotide, you have this position, this position, this position with the same nucleotide. Now, why should we bother about this? Why should we be concerned about identifying these mutations? Is it not easier to live our lives as if this did not exist?
Does that like one change help you understand what could lead to like possible changes in our, I don't know, like with the neuroblastoma, for example?
Oh, that's a very good question. That's a very deep question for neuroblastoma. Then the answer to that being that it's a very complex answer, but it's a very good question for neuroblastoma. I'm going to explain it in maybe 2 minutes. Neuroblastoma is a pediatric cancer, and pediatric cancers in general are thought to be less led by mutations than adult cancers. So in adult cancers, mutations are very important, I would say, because there is this concept of neoantigens in, well, one of the reasons is because there is this concept of neoantigens in adult cancers. So what I'm trying to say is that in adult cancers, it is thought that mutations have a very important role, including the mechanisms of aggression in the adult cancers.
Like
Now,
how aggressive the cancer is?
yes. So for example, including variants that have a role to make I'm just explaining this at the very high level. I need to understand this much more deeper to give you the exact details. So that I'm just trying to convey the general idea, right? So including the idea of this variant is more dangerous for an individual that is of Asian background. So in adult cancers, that's much more important than in pediatric cancers. In pediatric cancers, what is thought that leads aggressiveness, mechanisms of aggressiveness, are the epigenetic mechanisms that I did not discuss so much with you, but that's why I wanted to focus just a little bit on the hypoxia mechanism, because that's one of the mechanisms that is thought to be important in neuroblastoma. But coming back to this, yes, you're very right for adult cancers. You
Mhm.
are a little bit right for neuroblastoma because even though mutations are not thought to play a huge role in neuroblastoma, there are some mutations that are important in neuroblastoma. So in that regard, you're right. So I want to also mention, I don't know if you wanted to comment anything else.
Well,
Or
I was just wondering, it doesn't cause the cancer, right? Does it increase your like does it increase how likely you are to get that cancer? Or can it?
that question is a little bit tricky because certainly there is association between the variant and the incidence of cancer, but the fact that you have the variant does not mean that you're going to have the cancer. It doesn't mean that the variant is going to cause you to have cancer, right? There are other
You mean
factors. There are other factors that play into cancer development.
by looking into like these SNPs, would we be able to figure out like, oh, if this person has like maybe this mutation in this location, they're maybe like more likely or they're more predisposed to get a certain type of cancer?
Yes, you can definitely calculate risks, but the good thing about the risks is that they are never 100%, right? So there was the example of that actress, Angelina Jolie.
Okay.
Right? That was her case. There were, and I guess in her case there was something that was more aggravant. I don't know if there is a. Let me see what the word for that is in aggravating the fact that she had her mom and her aunts with breast cancer, right? That would be, I would say, an indication or a contributor to having a mutation, and that certainly has contributed to her decision on going breast surgery. Um, I would say I do not know. I'm not an MD, but I would say that the probability and the odds would be much more less against her. I would say if she did not have her mom or her aunts or sisters or relatives in a condition that is similar to that, right? We can definitely calculate the risk, but and it's good that you think about calculating a risk because you can definitely develop an algorithm to calculate that, but it's not going to be 100% accurate. It's not going to be 100% correct all the time. There are other factors.
All
Uh,
right. Thank you.
sure. All right. So that's why we want to consider mutations. And if you think, and so for example, if you consider things like COVID, like right, SARS-CoV-2, so it is thought that mutations in the genetic material of the virus has caused it to do the zoonotic jump from um, a mammal to humans, and that's what has caused the entire COVID-19 pandemic. So I think this is a very important concept, and the reason why, and one of the reasons why I want to show you how we can run a variant call pipeline. Do
Hey, Gio,
people?
another question.
Uh-huh.
So, like you're bringing up this COVID pandemic example,
Uh-huh.
would we be able to create algorithms through R that would allow us to maybe identify like which mutation, or like if it's caused by a specific SNP?
Well, the mutation is the SNP, right?
Yeah, yeah, yeah.
Yes.
So we would be able to create code or algorithms that locate that
What we are going to
mutation.
do, that's what we are going to do today, but we are going to use Bash instead of R. I
Okay.
think it's right now at least for me, it's easier to use Bash than R. And the good thing about the overall concept of this is that you can run COVID samples, you can run human samples using a pipeline similar to this. I think if you use it R, or maybe not. At least I was limited in the usage of R when I tried. But I do not think that I have, what is the word? Exhausted all of the possibilities in R. For me today, I think it's easier using Bash instead of R.
Okay, thank you. Sounds good.
So that's it for this part, for the theory of this part. I hope it makes sense. Do you guys have any other questions or comments? All right, so let's do one thing now. Let me close this. So I have these commands here. What I want the Mac OS users to do, I want you guys to try to follow these exercises, follow through them. You might not get the exact output as I did because I did this in my local machine, so you can see here that it's the Bioinformatics Bridge course folder. But I wanted to try this while I help the Windows users get their terminal. If you do not know how to do this in your terminal, let's including the Mac OS users, let's use the ID, the online IDE. I don't remember. I should have put the link for it somewhere here. Okay, so this is what you are going to follow. And then, so this was this. This is the variant called pipeline, correct? Oh, these are the exercises. Then you can also, we're going to work on these exercises here. Oh, here, there you go. These are the variant call pipeline exercises. This is the IDE that I want you guys use. We have used this before. So here, so for example, what I'm saying here is use the echo command to print a greeting to the class like this one. So for example, you can copy this, come here and paste it and run this. We did this last week, right? Oh, of course I should change it. Change the language here to Bash. I'm gonna close this and reopen it. And I'm gonna change this to Bash. And I'm going to come back here, copy this again. And I'm gonna run this. Why it's not allowing me? Okay. The internet is not very good today. Don't know what's going on there. So the idea here is for you guys, some of you, to follow through this. There are instructions here, and I can help if you guys need did anyone open the IDE, the online GDB IDE?
Yeah.
Did this command work for you?
Not really. Nothing's really showing up.
Yeah, let's give it some time. And then there are these exercises here. And then finally, we're going to access the AWS image instance pretty soon. I do not want you guys to do this right now. I'm going to create, I don't know what's going on with the IDE. I am going to create 2 breakout rooms, or maybe 3. Maybe we're going to need 3. I want so one is going to be Mac OS. Two is going to be Windows. And let's pair three. They're oh, it seems like Zoom split you guys, but one is for Bash, two is for Windows. So if you're a Mac user, use one. If you're a Windows user, use two.
Hey, Gio, would you be able to put me in one? It's only allowing me to join two.
Yeah, same right here.
Oh, really?
It's telling me to join room 1, but I'm window.
Kanav, you were sent to to room two, right?
Yeah, but I'm a Windows, so I should be in one.
No. If you're Windows, you're in two.
Oh,
And the option of choose your breakout room versus the join because
Etenish,
if anyone
where do you want to go, Etenish?
well, what's room 3 again? Because
No, room three is empty. It should be empty.
okay, all right. I was like randomly assigned to room 3. Can you assign me to the Windows room then?
Windows is two.
Me too, please.
Yes, I'm gonna. Why can I not send Etenish to room two?
I'm Nithya.
So I sent you. Sorry. Wait, Nithya, you wanted? Oh, okay. Eric, are you the last one?
Hey,
Victor?
Gio, I want to be in round 2, same as Nithya.
I sent you there, right?
Nope. I received an invitation to round 1. Oh
Sorry.
yeah, thanks.
Kanav, you want to go to one or two? Who else is here? Eric, which room do you want to join? Eric? Eric, do you hear me?
yeah.
And like to go to anywhere you can like do CD.
I think we should maybe break these people here into two other rooms because the three instructors are here. Let's stay one per room.
Okay. Is this the Mac or because I got Windows and Mac users here?
Wait, this is a room two, so it should be Windows, right?
Yeah. Okay. Kanav is
Oh, my
Mac.
fault. I thought this
Since,
was
you
Mac.
had randomly assigned people instead of choose breakout room, you're not able to choose.
I
breakout
think
room too.
I think that Zoom assigned randomly, and then I sent a number of people to a number of rooms. I do not know who I send where. I just try to follow what people told me they wanted.
You could assign me to a different room then.
Yeah. So if I give you guys co-host
We
privilege,
can assign people.
you can? Okay,
Yeah.
let me.
If you
let
give
me
us host
see.
privileges.
So it's the 3 dots. But I
Go
don't.
to participants and then more under our names.
It's not allowing me to. It's not allowing me. Maybe is it in the main
Oh.
room only?
I think we might have to go back to the main room to for you to give us the co-host thing.
I just made Nithya. Where's the finish? Oh, here. I just made a Tanish co-host too. I'll create another breakout room so that we can have one instructor per room with the Windows users.
I don't think I'm host. Oh no, I'm host.
Here, I can reassign it so that way it's the way you wanted it. Or oh wait.
Breakout rooms.
Think it really matters at this point. But yeah, do you want to open up your?
So
Were
here
you
you
having trouble opening it up?
No, Etnish helped me out. I got it.
Okay.
Here you guys are only going to follow the commands, right? Either in the IDE or in your own local machine, right?
Yeah, it looks like both of them have it set up and they're doing it independently.
They have not?
No, they have it set up and
Mm.
they're yeah,
Okay.
they're working on it.
I'm going to try to help the Windows people.
Okay.
Instead, no?
Huh?
Etenish, do you
Yeah.
want to try to help maybe one or two people in another room? Because maybe three people is, oh, there's only, yeah, there's three people here.
Yeah,
Three,
there's only 2 people
but do you want that I take
for
one?
Windows. Huh?
Do you want that I take one person because maybe three is too much, or one or two
Oh,
people?
Victor already set it up, so it's just me and Jose.
Oh
Okay.
yeah. I set it up with Victor last week.
Okay, Victor, can you share, can I try to help you in another, in the other room? Not that it's not installed, I just want to make sure that he can follow the steps.
Mm-hmm.
Okay, Victor, can you come with me to the room 3? Thank you, Atenish. Thanks, everybody.
Yeah.
Right here, is it? This room? Or room 3?
I think so. Let me see here. No, let's go to room 3 because this is the main room.
Oh, okay.
Let
Can
me,
you send me there?
yeah.
Because, yeah.
You hear me?
Yes, you can hear you.
Yes. Okay.
I share my screen.
Yeah.
Okay. Yeah. Can you see?
I can see your screen. Do you? What is this? Did you already install it?
Not. I download from the page that Etanesh sent, and I am installing right now.
So I don't know why you came back. What happens if you hit next and next?
No, no, I don't came back. Soon redirect me to this room.
Can you close this and open Git?
Okay. This?
No, no, no. There's an icon in the tab below. Open the terminal because this is
Where is it?
the icon in your screen down below
Okay.
here.
Okay. Yes, this is.
So go next.
Or can you see this window?
Yes. Hit next,
Yes. Okay.
next, 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 next,
Okay.
next, 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 next. Keep going. Keep
Okay.
next, 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 next. Keep going. Install. Let's do the defaults.
Okay. I don't know why why I'm being redirected.
You're not being redirected. You did not finish installing. Give me 1 minute. I need to go see my baby because she's crying so much. I'm gonna be back in 1 minute
Okay.
while this finishes installing. Oh, finish. Go
Okay.
click finish. Oh,
Okay.
yeah.
I'll wait for you. No problem.
No, no. Where is bash there? Where is git there? Búsqueda, haz la búsqueda
Okay.
de git. Git. Ahí, sí.
Git bash.
Git bash, sí.
Okay.
Sí.
Yes.
Haz un PW,
Ya.
PW,
ID.
sí. Enter.
Y enter.
Ahí está. Y entonces puedes ver el C, la C, users, José.
El directorio, ¿no?
Sí, sí. ¿Usted ha usado el terminal antes?
Ocupé esta, no, ocupé la de Windows, la que le dije antes, ¿no? Windows PowerShell, que igual
No.
es Bash.
Puedes y haz un PWD. Ok.
Ahí. No, está
Necesitas un
mal.
espacio.
Sí. Y el PW.
Yo,
Pero da algunos problemas, sí.
sí, porque yo no sé si este Windows PowerShell es el mismo del terminal. No sé, yo no estoy cierto.
Sí, intenté hacer los ejercicios antes con este de acá y me dio errores. Por eso mejor lo instalé este.
Ok, ok, pero yo pienso que es mejor usar este Git, pero
Ujú.
estoy un poco más cierto porque yo no soy un usuario de Windows, ¿sabes?
Claro,
Sí.
sí,
Ok,
está mejor.
déjame, solo necesito ver a mi niña y regreso en un ratito.
No, claro, no hay problema. La espero.
Okay, yes, that's it. Let me see if there's anyone else that needs help and if the other people have done. You can keep
Okay.
practicing, okay?
Okay, thanks.
People left here. Do they have their terminals working here?
Yeah. I don't know about Jose. He left before I could like help.
I
him
just was just
out.
helping him in the other room. He has.
Okay, yeah.
Did you
Then
help?
at Veta, I did help.
Okay. Did you help Jose with the PowerShell too? Windows PowerShell?
No, no,
Okay.
he left before I could help him out.
Victor Advaita, can you guys show me your terminal?
Yeah, sure.
Um,
Let me
yeah.
let me
Oh,
screen
I
share.
guess.
Oh, do you want to do it first?
No, you go first. I'll go second.
Okay, thanks.
Okay. Is this Git or Ming? Is it Git or no?
What do you mean?
Did you download Git?
Oh, yeah, it is. Yeah.
Okay. Can you do a PWD for me?
Yeah, sure.
Okay, ls. Okay. Can you do cd and then backslash?
And dot again, or just that?
No, just that.
Okay.
PWD now. Can you do just cd and enter and then PWD? ls, sorry. Okay. That seems to be your root user.
Yeah.
All right. Okay, so do you have any clue how to navigate your Windows system?
What do you mean navigate? Like, do I know where everything is?
Where documents and folders are located? For example, do ls users.
Yeah.
US. Okay. You see, you had that one folder before. You were in. It seems like you were.
Yeah, I was in 14084. Like that's where all my stuff is, pretty much.
Okay. Do you know where your desktop is located?
Yeah, I think it's 14084.
Do you mind going there and checking?
Yeah, sure.
I just
Oh,
want.
like my actual desktop or in the terminal?
Well, your desktop in the terminal should be your actual desktop.
Oh, yeah.
I would think that it should be inside the users, maybe inside all users.
Oh, yeah, let me look at
You
that.
need to know
Oh, oops.
where all your
I think.
this is the
I
main.
think it's over here, I think. Wait, yeah, my desktop is in my 14084 folder.
Yeah.
My
Can you
desktop's
not
right here.
find the 14084 folder?
How do I switch back to that?
Can you scroll down?
Yeah.
Do you type ls and enter? There's a 14084 folder not in there.
I think it's in users.
Oh, okay.
You
And
need
go
to
to
CD
users,
to users.
and then the 14084 folder should be in there.
Yeah,
You
it's
CD
in
into-
users.
Right, so CD, go to users, type in users, and then backslash 1484. There you go.
Okay, and then do I ls now?
Yeah. Well, you don't need
Okay.
to ls. You know
Oh,
your
yeah.
desktop is there, so you can just CD into it.
Oh,
I think yeah, it works. It works.
I think you can CD now to your desktop. Let's see. No, you,
Like CD desktop.
yeah. Tap.
CD
Wait,
lets you go into your another directory.
Okay.
ls just lists what's under the current directory.
Okay, I think it worked.
Can you PWD there?
Yeah.
Now I'm sure you're inside your desktop because the last one is your desktop there, right?
Yeah.
If you want, you can ls now and see if you recognize any name of any files there. This is recording, right? Okay.
Yeah, it's the desktop.
Okay. So that's how you know where you are located in your machine. That's good. I needed everyone to have seen this. So everyone is back here, correct?
Can I stop sharing or do you want me to
Sure,
keep
yes,
sharing?
yes. Thank you very
Okay.
much, Lita.
Yep.
Okay, who else has doubts about how they can locate the terminal in their machines? I know that Jose has it. Victor apparently has it. Kanav, I don't remember seeing your screen. Do people want to share their screen and that we try to help them identify the terminals in their machines? Okay, I'm going to share the screen again. So let me know if you can, just like me, just like I am going to do now. I'm going to open my terminal here. You've seen my screen, correct?
Yeah.
Okay. I am going to open my terminal and here is my terminal. I'm going to put the terminal screen here on the side. So I can, if I do echo print a greeting to the class, it prints that. And so this is what this line is, right? And now print your greeting message. So you need to insert the message using the echo command. So the way to do this, so we're going to type echo, and then you can say hello, but I need to switch. Or I can say hello, hello class, hello everyone. Another thing that I can do is I can create a variable, right? So I can do test underscore variable equals type this file. So if I echo test, it auto completes because in the bash environment, the variable was just created here. So I can echo what do you think is going to be the output of this command, Victor? What do you think is going to be the output of this command? Victor, do you hear us? Pranav, do you have any guess about what we are going to see here?
it would output this file, this underscore file.
No, what the output of echo $ test underscore variable, what the output is going to be.
Yeah, wouldn't it just output this underscore file?
Oh, yes, sorry. Yes, yes. So this is the variable. The way that I understand it is that this is assigning a value to the variable. So when I echo, it echoes the value of the variable. Thank you, Kanav. And then if I ls here, it lists a bunch of stuff for me, and you can see that these, whatever these objects are pretty different than the results that I had here, right? Because here I was inside, when I wrote this, I was inside a folder that is called exercises. So I'm gonna echo this command. And the result of this command was list the files. These are the files inside this directory, and this is what the output of that is. I'm going to give you maybe 5 minutes to see if you guys can locate where you are in your computer. That's a very important thing, because what we are doing, what this is training is your notion of path. And by the way, we should do a 5 minutes break right now because it's 11 past 7. So let's do the 5 minutes break, and then let's continue from this part here. And I cannot highlight how important it is for you to figure out how to navigate the terminal, knowing where you are located. Okay, let's do the break, and then in 5 minutes, I'm going to discuss a little bit my location inside my computer. Hey, Eric, we are in the 5 minutes break, but we are here in this step. I'm going to talk a little bit about finding your location in your computer in the terminal. We are in the terminal commands and variant call pipeline. It's this one here. And here I am in exercises and assignments by dates. All right, everyone. 5 minutes have passed. I'm going to share here again. I'm connecting right now to the AWS image. Do you guys see my screen? Do you guys see my screen?
Yes.
Okay, thank you. I'm connecting right now to the AWS image. So I'm already inside the image. I'm not going to bring you guys here right now. I'm gonna bring you in a few minutes. The reason why I'm showing this is because I need the user. And this is what the user is. And I'm going to paste it. I'm going to put it in the chat. I'm going to actually put it in the chat. Do not SSH yet. I'll tell you what to do with this thing that I put in the chat in a minute. Okay, but we were here and I was going to explain about the directory, right? So if I come back here in my tab, if I print working directory, it's telling me it's this users / Gipoliano Chavez. And if I list again, there are these folders, there are applications, Saros space configuration, desktop, and so on and so forth. So for me, I always know that in my computer, and for some of your terminals too, this tilde and then the slash is what is known as the root folder. And I know that that my desktop folder is there. So this desktop here is located inside my root folder or my root user. And so if I change my directory to the desktop, and if I pwd, I'll see that I'm inside the desktop. So if I list whatever is inside my desktop, there are these files that have these weird names. So I recognize the folder that I have here because I do have a Gipoliano folder in my desktop. I do recognize the script.docx because I do have a script.docx in my desktop. So I know I kind of have an idea about where I am located because I know how to locate my desktop in the terminal here. And so if I type this command there, I am not going to locate these files right now there because I do not have these files where I am at here. So if I paste it here, it's going to give me these results, but now it's giving me these results because I know that I'm inside my desktop folder, okay? And here I was somewhere else where there were these files. So you can do the pwd to see where you are located. You can also create this make directory. So for example, if I type this in my terminal, and if I come back here in my desktop, I just created this test directory there. Were you guys able to locate where you created this test_ directory? Yes or no? Do you know where you created the test_ directory?
Yeah.
changes.
Okay, good. That's a good sign. So I created these. I created this directory. So if I run this command again, it's going to give me an error saying that the file already exists, right? Let me just take the attendance here. Okay. So I can move into that directory, right? I can change the directory to the test directory. And I can do pwd. And now you see that I'm inside this test directory. I did the pwd. I can also ls what's inside of there. So I need to explain to you that if I copy this command and type it in here, it gave me an error. It gave me an error because I'm inside test directory, it cannot change directory to that. The reason why I wrote this line here is because in the notebook that I got this from, this is actually, I cannot show you that this is actually a HTML file. In the notebook where I got this from, I have to, this is a mandatory command for me, not for you. So we can also run this. This is a little script, okay? There are several commands here. So it goes to, because I'm inside test directory, I'm going one level up. So I'm going to copy this, all of that here. So I'm inside test directory in ILS here. There's nothing. So that's why I put here, as you can see, there's nothing inside this directory. Because there is nothing here, let's create something in the next part below. With this command, we can create 3 files inside the test directory. First, I want to make sure I am inside test directory, which I am. So because I am, I do not need to cd there anymore. And I already did the pwd. So I'm going to copy this first line here. Touch first file. And if I list, I'll see that I created this first file there. So I'm going to copy the next two lines that contain the touch, and then the third line has the list, and it should list the files the first file, the second file, and the third file. So you see, if I ls again, it's giving me first file, second file, third file. Let's have the terminal print the names of the text files in the test directory for us. So I'm gonna do that again using a for loop. Please take a very careful look at this for loop, okay? So I'm going to copy this line. I'm going to copy this line. And I'm going to paste it here. Can anyone try to explain to me what this line just did? Can anyone try to tell us what this line just did? It is important to try, guys. So you can see here that it printed to the screen the name of this file is, and then colon. You can see the syntax that originated that output, right? This is the for loop. So for I in everything that ends with txt. Oh, I had never seen this. For I in everything that ends with txt, do echo, which we know what it is. The name of this file is, then colon, and then that thing, and then done. So for everything that ends with text, print the name of the file is, and then $sign I is the name of the file. And the reason why I put this $sign here in front of the I is because I just created the I in the for loop. And this I is created contrasting the I itself with everything that ends in txt. Because what is everything that ends in txt? Everything that ends in txt is each one of these text files. I hope it makes sense, or some sense. If not, if you want that I repeat it, I can repeat it. Okay. So it printed for us the names of the files. It printed what were the files, right? So let's have the terminal print 1 word, 2 words, and 3 words to the files like this. So we go to the test underscore directory, we do pwd to make sure that we are inside test directory. I'm gonna do pwd here. I do pwd all the time because I never know which folder I am inside, so I need to do that. So we can still list the files there. So what is the command that I can use to, does anyone know what is the command that I can use to see what is inside the file? Does anyone know? So that command is cat. We should see this command again somewhere. Well, ls, Alexander, thanks for answering. ls is going to list what is inside a directory, but cat is going to allow us to concatenate what is inside, for example, the first file. So cat first file is going to print what is inside first file.text.txt for us. So if I hit the return here, it's going to return nothing because there is nothing inside that file. Now, let's run this first line here. I'm gonna do echo word 1 and then greater than first file. Does anyone know what this, does anyone have any idea about what this is doing since I already told you here? Any ideas? Yes, the words are going inside the file. So the first line is echo word 1, go to first file.txt. So I'm going to copy this. Remember that when I typed the cat the first time, it showed us nothing. So now we expect that it has the word 1 word there, right? So if I go back, if I do the upward arrow and go back to cat first file, that's it. It has word 1. And if I do, oh, so in these 3 lines here, I am doing the same thing for each one of the 3 files, right? So you now know what cat does. What this star is going to do there for us. Remember that here in the for loop, remember that we have these 3 files in the folder here, right? When I use the for loop, I said for I in everything that is txt. So the star means everything, correct? So any idea what this command is going to do? It's going to cat everything, right? So I'm gonna hit enter now. Does it, can anyone explain what is this? This is the result of catting each one of the files. Yes, it's the words in each of the files exactly. The reason why they are equal here is because this is coming from first file, from second file, from third file.txt, right? And now we are going to echo word 2 only into the second file and the third file. We are not putting word 2 into the file, to the first file. So I'm gonna do that now. And I'm gonna do that now. So let's inspect the first file. It has only word 1. Let's inspect the second file. It has words 1 and 2. Let's inspect the third file. It has words 1 and 2. Let's now do the last line here. So let's inspect again the first file, only word 1. Let's inspect the second file, words 1 and 2. Let's inspect, oops, second file. Let's inspect the third file, words 1, 2, and 3. So does it make sense that we sent word 1 to files, first file, second file, and third file? We sent word 2 only to second file and third file because they are here. And we sent word 3 only to third file because it's only here. It's not shown in the second file or in the first file, right? Okay, what can you guys think about the order that the terminal is appending each word to first file, second file, and third file? What is the order? Well, every time it does the bigger than command, it sends the word to the very end of the file, right? So, for example, let's do for the third file, I'm gonna do a word 4 here. And if I cat the third file, the word 4 is there. And then if you do word 5 and cat the third file, although it's the third file, it has 5 words. Now, so that's what it does. It appends a word to the end of the file every time. Notice that I am using 2 bigger than signs here, or 2 bigger than operators. Does this operator has another name in English? Sorry. But okay, so that's it. So you use these 2 operators, these 2 symbols, to make the word go to the last line. Now note that if you use, for example, word 5 to third file, let's see what happens. Third, it erases everything and only maintains the word file output or input, right? I'm going to actually, I don't know, I wanted to put it in my source code, but seems like I cannot. Okay, and that's what I just did, this part. I cannot highlight how important the for loop is for you. So, or at this point, for good or for bad at this point, if this does not make sense to you, the only way that you're going to learn it is repeating it, practicing it. But I think if you are from computer science or from any of the engineerings, I think this is a good thing to practice or to think about. Okay, so in the for loop, what we're going to do is we're going to, well, this part here concatenates what's inside each one of the text files, right? So the output here was word one, word two. So this is coming from first file, correct? This is coming from second file, right? And this is coming from third file when I ran it here, but here it is different. So this is coming from first file, this is coming from second file, and this is coming from third file, because remember, we used this operator instead of this operator here. And so it replaced everything that belonged to the third file and replaced it with the word 5 that I just echoed here. Now, this is a command that we can use to count the lines in each file. So we can, so this is word count and then an option. L means line. And so how many lines were, not the first file? Does anyone remember how many lines the first file is going to have? 1, right? Yes, very good. How many words the second file? 2 and 3. I don't think it has 3 lines anymore because I broke the protocol. I broke my own script here. It has 1. It has 1 again because we replaced everything that was inside of here with word 5. Yeah, 1. Exactly. Okay, that's it for this part. And so with PWD, we are inside the test directory. I'm going to go back. I'm going to go one level up. Okay, so if I ls here is my test directory. And what I'm going to do now is I'm going to do rm minus r and then test directory. And I'm going to remove this directory. So if I hit this, I'm going to lose test directory forever. Okay. So if I ls now, I do not have the test directory there anymore. We need to be very careful with with this rm command because it means remove. And minus r means remove it if it's a directory. And we just removed our test underscore directory with this command. I think that's it for this part. Did anyone have any problems to follow along? Did people try to follow along with me? Because if you tell me no now, I will not know if you guys have any problems to connect to the terminal. So, okay, Alexandra said it worked fine for her. Did it not work for anyone? Was anyone not able to follow anything? So I'm assuming from this point on that for the Windows users that I was much concerned. you guys can use the terminal with no problems. Is that correct? Okay. All right. So let's move on. How much? We have only 20 minutes left. Oh my God. I'm gonna close this. I'll let you guys do the variant call exercises. These questions here. We don't need to do this together now. But please do that. Now, I guess it's time to get started on this. So in your terminal, I'm actually going to close my terminal and do, I close at least that one tab. And now I'm going to open a new tab. I'm going to open this new tab. In our terminal, you're going to SSH and then the DC user and then the name of the address of the instance. Okay, so you can copy the line that I put in the chat. I'm gonna put it again. I have to copy it back. So you can copy this line. You can copy this line in your terminal. And I'm gonna show you doing it from here. You're seeing my screen, right?
No.
No?
Nope.
That's why it's always good. It's good to keep asking. Okay. Here we are. So everyone was able to copy this command and put it in the terminal. Do not hit enter yet, because and if you did hit enter, there is this data for car password that is needed. Do not do it yet. Let me just walk you through this. Please note that I did this as questions. Okay. To guide you through this. Once we enter the terminal in the instance, we are going to copy, you guys are going to copy the variant call folder that is in there with your name, just like I did here, because you are going to run the variant call pipeline inside your own folder. So this is the variant call pipeline. And you can see here that I put a reference. I just need to fix this syntax. I put a reference here to where I got this syntax in the code notebook. So you can see where I got this from, right? We were just discussing the variant call pipeline and so on and so forth. So I put the link there in this question here. And so the syntax for this is we change the directory to a FASTA directory. I was saying that we are going to use one of the mitochondrial sequences as the reference. What is this? Does anyone know what is this? What are we creating here? By the way, we just discussed this a little bit ago. We are creating a variable, okay? We're creating a reference variable. And this is going to contain the FASTA file information. And here is the for loop again. And we are going to do 1, 2, 3, 4, 5 5 tasks. We're going to do 5 tasks in this for loop. 1, 2, 3, 4, 5. 5 tasks in this for loop. And I need to explain to you. The first task that we're going to do, this is I think this is called sliding the string. We are picking only the first 8 letters of the string because I only want to take this part of the name. Okay, I only want to take this part of the name. Once we have slided this string, the FASTA file, remember, for FASTA file in everything that starts with AF and then ends in FASTA, because I did my math and I just discovered that the names of the FASTA files, they only vary in this region of the string. That's what sliding the string means, or cutting the string is another way to maybe help this make sense. So for FASTA file in everything that is called, this is how the computer understands, right? Everything that is called AF star.fasta do, then I define my other variable, the FASTA file name, and this FASTA file name is a fraction of the FASTA file that is in here. And remember, the names of the FASTA files look like this. By the way, I'm going to show you what the FASTA folder looks like. So we are talking about, oh, I did the SSA. I'm going to enter I'm going to hit enter. You guys can also put the password there. The password is data for capital C ARP, data for carp. I'm going to hit enter here, and I'm going to come back here and show you that. So I said here that once asked, type the password data for carp. This is what the password looks like, okay? So I came here and it printed this message here, welcome to Ubuntu, blah, blah, blah. And then it said last login, some sort of date. Apparently it was September 10. Apparently I logged in from the future here. So I do a PWD. So I'm inside this thing called home and then DC user. DC stands for data carpentry. So I'll do a PWD again because I managed to forget where I am. So I'm going to list again. Oh, I didn't list yet. So you can see here the variant underscore call folder. Do not do anything here. Do not do PWD. Do not do ls. I'm just showing you what is inside the variant underscore call. So I'm going to do ls variant underscore call. And then if I hit enter, it's telling me that I have this bunch of folders there. I have the BAM, the BCF, the FASTA, the references, the SAM, the SNPs underscore VCF. So I'm going to go back to my ls variant underscore call. I'm going to go to FASTA, and I'm going to ls there. You see that I have a bunch of .fasta files. Apparently I have 1, 2, 3, 4, 5, 6, 7, 8, 9, 10, 11 times 3, 33, plus 1, 34 FASTA files. 34 FASTA files. So I was just showing that because I was going to explain that. So you see here that at some point, we're going to change the directory to inside that FASTA file, that FASTA directory, right? And so we're going to run the for loop as for FASTA file in all the 34 FASTA files in here, that for loop is not going to look at, oh, it's actually going to look at this FASTA. This may give us a problem or not. I am not sure yet if this gave me a problem, but only now I realized. So that's one thing to keep an eye on. But this file here is not going to give us any problem because it's a .txt and I'm running my for loop based on the .fasta. Okay, so for everything that has this pattern, oh, but I see, so we should not be concerned about this because it does not have the AF pattern before the star. So you see that all the other 34 files do have that. Oh, and that might be the reason why I put AF star dot fasta and not star dot fasta. Now I think I remember this. So for everything that is AF star that has the pattern AF star dot fasta do, then change the name. I'm not going to bother so much about this. And then the next thing that it does is it aligns, okay? It aligns and then it produces an alignment that is sent to a folder called SAM. So the SAM folder, I can ls. So ... means something that is above the current directory. The current directory is the FASTA directory. You can see that. So here in this line here that I'm highlighting, I am inside the. No, I'm not inside. I'm lying actually. If I do, that's why I have to do pwd all the time. I am still inside DC user, right? So if I ls, I see that the variant underscore call is the one folder that I'm talking about. So inside the variant call folder, there is the SAM file. So the SAM file is where the alignments are located, right? So in the variant call, there's nothing there because you guys have not ran the variant call yet. So that's it for the alignment. And then I'm going to convert. We are converting the SAM to BAM. So both SAM and BAM are alignment files, but the BAM is binary, so it's less heavy, and that helps a lot when you deal with several files. And then, so up until here, we have, I'm gonna say processing, because in this step, in the next step, and in this step, we have the identification of the variants. So what the next step does, I'm going to use the word piles here because we are going to use the reference FASTA file, to detect or to compare the alignments, and in this case it's the BAMs, okay, the BAM sequences. So it compares the BAMs, right? The BAM is an alignment file to the reference, and once it is comparing the alignment to the reference, what this command does is it detects variants. But remember that the BAM file is a binary file, so the BCF is also a binary file. Now what the next step does is it extracts the exact type of mutation that we want, that is the SNP mutations, okay? So in this step, we are also using VCF tools to extract SNPs from the .bcf file that we just produced. Okay, so I'm going to run this very quick because we only have 4 minutes left. We do not need to do anything very different than, so let's do this. Let's everyone copy this line. Oh, first of all, did you all get to go inside the DC user folder? That's the AWS instance. Did anyone not enter the instance? Alexandra's there. Actually, I'm going to. Okay, I'm going to variant Booleano. I'm gonna make it variant Booleano 1. Okay, let's copy this command. So we're going to copy the variant call with our own names. And so I'm going to write here my name. That's why I saved my _1 folder with the number 1, because now I will have a folder here with number 1 and another folder with my name that I'm going to run right now. Did people do that? Nobody? Are you guys having any problem to copy this line here? So you copy it here and remove my name and put your name. Did anyone try in doing that? Kanav, did you try to copy the variant call folder?
Yeah.
But why? I should see it here. Do you when you are, where did you copy it?
Here, let me try again.
Can you share your screen?
One sec.
Sure.
Oh, shoot. I just closed out of my terminal. One sec. Uh, okay. It just kind of restarted, but yeah, I'm here.
Okay,
Do
can
I
you?
need to come back into it?
Yes, you need to SSH again.
Okay. I think I already have copied. Yeah.
Please, one moment. One moment.
I think that's not the correct IP.
Yeah, it's not. It's the one that I have in the chat.
Okay, let me try that one.
I put it right now. Jose, were you able to enter?
No, I can't. I couldn't, but I paid attention to your explanation. I think could be my IP location that
Oh,
it
okay.
cannot be possible to me for access.
I'll take a look at your screen in a minute. Kanav, I don't think you connected.
Here, I'm trying again.
I see
There we
Alex.
go.
Just created her object there.
Hey, Gio,
Mhm.
why is it not letting me put in the password?
Can you show me, can you show your screen?
Am I, oh, whoops.
Oh, that's where you write your password
It's just data.
for capital C carp. Permission denied. Be careful. Hit the upwards arrow again. Enter. Now the DATA at capital. If you copy, what? Do not copy.
There we go.
Great. Okay. So once you are there, Kanav, so you copy this line and you come here and you remove Gipoliano and put your name. Jose, do you know that I try seeing your screen and see what's going on?
Okay.
Kanav, and try to copy a folder of the variant call for you.
Okay.
Jose, you can share your screen. Okay.
Yes.
Yeah, I got mine in.
Okay, great, Kanav. Hit enter there, Jose.
Oh,
It's
right.
DA. It's the same thing as Kanav. DA
Okay.
for carp with capital C.
I can write anything.
No. It's writing and you do not see.
Okay, okay.
You need to
DA.
DATA at capital C ARP.
Okay. I'm
You might have
writing
typed it in incorrectly.
DAC.
No, it's data for carp. I'm gonna put it in the chat.
Okay, okay, okay.
I wrote it in the chat.
Oh.
Do not copy it from the chat to paste. The IP changes all the time because while it's on, I am paying for it. So if you guys need to access it again, you need to let me know so that I can turn it on for you guys, okay? Because I'm gonna turn it off after class and the IP is going to change again. All right, Jose, great. Can you do PWD and MLS? Okay, Eric, thank you. See you. And
Okay.
this is going to be recorded. If you can watch it. Right. And then, okay, let me show you Jose again. So what you do is you copy this line, you go to the terminal. So we are inside the DC user ls, and then you paste that one line here. You remove my name and put your name.
Okay, I'm going to do it right now.
Kanav, you need, do you know which variant, which folder you want? Kanav, do you know which folder you want? Because you have variant underscore cal underscore Kanav and variant underscore cal Canav, which one do you want to maintain?
The one which has like the underscore. I actually forgot the underscore in the second one.
Okay, so do you want that I remove this one?
Yeah,
This one, right?
yeah, the one where it just says call and then my name right after.
Without, oh, okay, so it's not this one, it's this one. Correct?
Yeah.
Okay, and then I see Jose is there. Okay, all in theory. Okay, we can copy the first line because the first line here in the variant call pipeline, the first line is changing the directory to the FASTA file, okay? But notice that you need to modify this user, correct? Not this user, this folder, right? So let's copy this, paste it here, and put your, for me, it's okay, it's already there, but put your name there. So for Alexandra, it's Alex. For Jose, it's Jose. For Canav, it's Canav. Canav.
Yeah.
Did you fix it? Oh, okay.
Thought I did.
Yeah, you did. You renamed it?
Yeah.
Okay, so for me, it's going to be this. So I'm going to hit enter now. So I'm going to PWD. I am inside variant underscore call underscore Gipoliano and FASTA. And if I ls, there's a bunch of FASTA files there. When you guys ls, do you see 34 FASTA files too?
Yes, and the last one is the same as you, sample IDs.
Good. So I'm gonna do one thing now because I want to do a sanity check. I'm going to ls and then // references. Try this command because I want to see what's inside references. So do you see something like this? So it's AF243627.FASTA. And the last one here is something that ends in .fasta.sa. Do you see something like this?
For me, yes.
Yeah.
Canav?
Yeah.
Okay, perfect. It means the variant call should work. You can now copy this line all the way to the bottom and paste it here, and it should work. So it's doing, did this thing show up for you guys? Did this output show up for you?
Yep, is something like processing.
Okay. Alex and Canav?
Yeah, I think so.
Okay. Canav?
Yeah.
Okay, great. It means it worked as it should have worked. So what I was going to say is that what that did was it aligned, it converted the SIM to BIM. It did the variant detection in these two last steps. The last thing that we're going to do is we're going to ls the snip underscore VCF there. We can hit enter. You see there's a bunch of VCF files there. Let's take a look. So remember that the star means everything, right? So if I ls star, I am listing all of them. So let's do cat for concatenate. Remember, I want to look at inside each one of the VCF files. So cat and then // snips underscore VCF /*. Let's hit enter. Do you see something like this? Each one of the lines is a mutation, which means each one of the lines is different compared to the one reference file. Do you guys see something like this?
What's the command again? I didn't get to fully write it down yet.
It was cat // snips underscore VCF /*.
I got the same thing that you did then.
Okay, Canav and Jose. Victor, I don't know if you had a chance to see everything that we did. I'm going to upload the recording today or tomorrow, if so you can have a chance to watch. And then I'm going to ask Alexandra or Canav or Jose to maybe help you with what we did here if you need help. Is it okay?
Yes, yes.
And then you
Yes.
can also email me or send a Slack message and I can try to help. But this, I'm going to upload the video to the website. But everyone, basically what we did here was we identified mutations. Now that some of these mutations in mitochondria, they may be correlated, they may give more risk, as Kana was asking, for these people. They may confer increased risk, for example, of Alzheimer's, of dementia, but that's something that we are not going to have a chance to look at. Basically, what my goal is for you is to show you how to run the variant call pipeline, which we just finished. Now, if you want to explore this more, talk to me for the purpose of your presentation on Friday, and I can help more on Wednesday. Okay? Questions or comments?
I won't be able to attend on Friday, so do I just like record myself with the presentation and email it to you?
Sure, yeah. And if you want, I can help you on Wednesday for your presentation and for all of the material that we covered so far.
All right, thank you.
Thank you. Questions or comments, everyone? All right, does it all make sense to you?
Yeah.
Okay.
Yes.
Okay. I am going to turn off the instance now, so you're not going to be able to access it. If you need to, just let me know. I can give you a couple more minutes right now, or I can turn it on anytime that you want. I just will not let it run overnight because I'll have to pay for that. All right, if you guys do not have any questions or comments or anything, that's it. Thank you so much for joining. Enjoy the rest of your night.
Thank you, Gio.
Goodbye. Thank you so much, you.
Thank you.
Oh, Mr. Gio, sorry, I had to like leave like I think last minute. I'm still like in the Zoom meeting because I had to do like like bridge. So they're making me do a meeting right now for writing.
Uh-huh.
So will you have like office hour? Like maybe you said Wednesday you'll help with the presentation. or try to help?
But Wednesday in the regular class, I'll help everyone with the presentation on Wednesday.
And that one's a little bit more, it's a little bit more different like than the other presentation, right? It's
At the I am not going to force people to present or on figures or anything. I would like people to present on the figures that they plotted. But if they want to stick with the commands, that's fine. That's fine. I'm not going to force anything.
oh, okay. No, I was just just asking because I thought, because there's a final project and I was just like looking at that and like, are we gonna do that?
Wait, you
Or
want to
is-
do a final project?
No, I was looking at that. I was like, is that what we're gonna do for like this last week? Do that final project presentation?
Well, the presentation is the final project presentation because it's a presentation that should contain everything that you worked on during the course, or it should summarize everything that you worked on during the course and the things that we learned.
Okay.
Yeah.
And wait, so there's no, you only you'll help during class, but like, do you have like, office hour you can still like help or?
I can help, but I already held my office hour that was today. If you need
Oh,
to
okay, I
meet,
see.
I can arrange a time to meet with you in a different time than the class, the regular class. I can
Yeah,
make
okay. Yeah, okay, that sounds good then.
okay.
Okay.
Just let me
Thank
know which day and time is best for you.
Okay, thank you, Mr.
All
Gio.
right.
Bye.
Bye bye.